right, guys. Today we're going to start a new chapter. This new chapter is entitled The Ways of Change. It's kind of an intriguing title. What is this title about? What does this Ways of Change mean? Basically, what we're going to talk about in this chapter is a lot of genetics because remember that in order for a species to evolve and change over time, what needs to change ultimately is their genetic material, is their genes, their DNA. So today that's what we're going to talk about. And before talking about mechanisms, we're going to understand what is DNA, what is this molecule, that is the molecule of heredity. We're going to define what is a gene and what is an allele as well. We're going to talk about this pretty soon. We're going to define some terms such as genotype and phenotype. We're going to look into different types of cell division, mitosis and meiosis. And then we're going to understand what are the sources of genetic variation in sexually reproducing organisms. We're going to have to know this really well. And ultimately, all of this, all these points are in preparation to understanding how allele frequencies behave in populations. Remember that here in this course, what we really aim to understand is how evolution works and therefore how populations evolve. So we're not really interested here in understanding what happens necessarily just in the single individual, but we really focus on what understanding of what happens in entire populations, what happens in genes that flow through entire populations. And so, again, in order to understand this, we need to know very well all the previous points. All right, so what's DNA? DNA stands for deoxyribonucleic acid. It's an acid, it's an acidic molecule. It's very large and complex, and it's found in all living cells as well as in viruses that actually are not really considered living organisms. So all organisms pass this molecule of DNA to their offspring when they reproduce. Here is a representation of the molecule. The molecule is a double filament, and it forms a helix. It kind of coils into space to form a helix. What are the components of this molecule? This is a large molecule that is composed by smaller components, which are known as nucleotides. Okay, the nucleotides that compose DNA are only of 4 types, and they are named as adenine, cytosine, thymine, and guanine. These are the 4 nucleotides represented here. Of course, I'm not asking you to remember the formula whatsoever. For this course, for the scope of this course, just remember we can compare these 4 nucleotides just to 4 different letters. Now, if you have 4 different letters, and imagine to actually make up words that are thousands of letters long, now you can understand how many possibilities you have there. So with these 4 different letters, you can make many thousands, hundreds of thousands of different words if you are able to put in each word like thousands of these 4 different letters. Remember, letters can also be repeated. They can be repeated many times, even 20 times in a row. Okay, so sorry, there is a little typo here. Okay, just discard this. You can understand the potential of obtaining many different words with just these 4 letters. Okay, that's the principle that you have to remember. Now, in eukaryotic cells, which is the type of cells we're going to focus on, because these are the types of cells that compose animals, Remember that DNA is organized in a structure known as chromosome. And each chromosome has two strands of DNA that are twisted into a double helix as shown here. Now, each species, from protists to the most complex of the animals, has a characteristic number of chromosomes. And you know, human species has 23 pairs of chromosomes. Each other species of the animal world, for instance, I can tell you mice have 20 chromosomes. And if I'm not wrong, dogs have 39 chromosomes, a huge number, okay? And so the higher the number of chromosomes, the higher can be the variability ensured in the species. And you will understand this when we talk about cell division. All right, so zooming into a chromosome of a eukaryotic cell, we will find out that the double DNA helix of which the chromosome is composed is actually not the only component of the chromosome. There are also special proteins known as histones. Actually, you don't have to remember this word necessarily, but remember, eukaryotic chromosomes are not composed by DNA only, but also have a very important protein component that kind of gives structure to this chromosome, which is really a little body of condensed DNA that you find at the moment when a eukaryotic cell needs to divide. It's a very convenient way to group together all the different chromosomes, to condense them together and make sure that no errors in terms of splitting DNA into daughter cells when a mother cell is dividing, no errors occur. All right, so when we zoom into a chromosome, which again, we can just focus at this point just on the DNA. Here there are no histone, no proteins represented. Each chromosome contains many thousands of genes, okay? And a gene is a functional region, as we will see. And what really these genes do is determine, they're like the code for determining the identity of proteins. So remember, genes in your body are important because they determine the correct identity of a protein. They are like unique words, and these unique words dictate the sequence of unique proteins. But as much as that is true, remember that each of these genes does not really have any function. It does not do a job. It's just a code. So what really does the jobs in your cells, in cells of your body, are proteins. And the identity of proteins is just as important as the identity of genes. If also proteins, just like genes, are made of units, sequence of units, they're called amino acids. And if you change the identity of a unit in this protein, this could be the consequence, a result of a mutation in a gene, then this could be very deleterious, could change the identity of the protein to the extent where the protein doesn't work anymore. So proteins need to maintain their identity to be able to perform the specific functions that they are dedicated to. All right, so for instance, let's look at some examples of proteins. Proteins can be structural. Remember, we talked about collagen. Collagen is the protein, the unifying protein for the animal world. Remember, we find it in sponges, and from there it's present in all animals. It's a structural protein. It gives a structure to the body of an animal, even to the body of a sponge to some good extent. So that's its job, to be like a column that sustains the body of an animal. But proteins can have other functions. For instance, insulin is a hormone that it constitutes a signal to lower glucose in the bloodstream whenever you eat a meal. Fibrin is another example of a protein that is involved in blood clotting. So many functions are performed by proteins, even defensive functions. For instance, your antibody, your immunoglobulins are proteins, okay? And so, again, insulin has its identity. You cannot change the identity of the gene that encodes for insulin. Otherwise, insulin may be altered to where it doesn't function anymore. Okay, so this is as important as, again, maintaining identity of a gene. All right, now, again, as I told you, we're interested in understanding how gene flows through populations, what happens to genes in populations, not necessarily in single individuals. And in populations, even in human population, for any gene, there are different variants present. I have brown eyes, and maybe some of you listening right now have blue eyes. So some of you may have hazel or green eyes. So these are all variants. The fact that I have brown eyes doesn't mean that I have a mutation compared to a person that has blue eyes. It's just a different variation, okay? It's a different protein that protein sequence and gene sequence that is just present in a population. And there are many examples. Another example is a skin color and so on. So all these variations in a gene, in a population, are called alleles. You have to know this definition very well and remember the definition of the word allele. Otherwise, you don't understand what I'm talking about if I give you a question, a specific question in the test. So remember, alleles are just variations or in a gene, or we can call them also gene variants as well. Now, the other thing that you need to understand is in terms of allele or these gene variants, there are alleles in populations that determine the appearance of trait in the heterozygous state. Heterozygous means when you inherit different alleles, one from mom, one from dad. If these alleles happen to be different, for instance, if I have brown eyes, I may have maybe inherited an allele for blue eyes from one of my parents. I may or may ever know this. It depends. So the variant that the gene variant or alleles that determines brown eye color is a dominant allele. So when it's this means that in heterozygosity, when it's present with a different type of allele such as blue eye allele, it dominates and therefore manifests itself. So a person has brown eyes doesn't mean that has two alleles for brown eyes. It could have one allele for brown eyes and one for blue eyes, or one allele for brown eyes and one allele for green eyes, because green is also non-dominant. And talking about alleles that are not dominant, that are masked by dominance, these are known as recessive alleles. So a recessive allele is an allele that is masked by a dominant allele in heterozygosity. So whenever a person inherits two different alleles from mom and dad, and one of these alleles is masked from the other, the presence of the other, and does not manifest, we're talking about a recessive allele. Dominant alleles are, by convention, they are indicated by capital letter, while recessive alleles are indicated with lowercase letter. Remember this. So it sounds complicated, but it's not. The other definition I wanted to talk about is genotype. A genotype is the allele combination that you inherit from your parents, or an organism inherits from the parents. Because remember that you inherited a set of chromosomes from mom and a set of chromosomes from dad, and each set of chromosomes brings its own alleles. Now, it can happen that mom and dad have the exact same allele in their genotype, but it's possible that they have different alleles. And so, if you inherit different alleles from mom and dad, then for that particular gene, you are a heterozygous. While if you happen to inherit the same type of allele, the same gene variant, then you are a homozygous for that particular gene. For instance, let's consider one very easy to understand example. Let's consider the eye color. Before moving on, I also need to define another term. Remember, genotype is the actual allele combination that you inherit from your parents. A phenotype is what manifests, okay? Now, a phenotype, what is manifested, does not necessarily coincide with your genotype. It depends. For instance, let's look at a person that displays a blue eye phenotype. Because blue eye color is a recessive allele, whenever you are in front of a person with blue eyes, you know that that person has a homozygous phenotype, which is of this kind, lowercase b, lowercase b. Remember, the recessive alleles are indicated with lowercase letters. And because if this person, in fact, had a dominant allele for brown eyes, it wouldn't manifest the blue eyes. So whenever a recessive a trait, a recessive allele is manifest through phenotype, you know the genotype right away. Whereas for a person that manifests brown eyes, has a brown eyes phenotype, such as me, this person, you don't know if it is, what kind of genotype it presents. It could be an uppercase b, lowercase b, because if he had the gene for blue eyes, the gene for blue eyes would be masked. So by looking at the phenotype, you couldn't tell if this person, in fact, has that allele. Or it could be uppercase B, uppercase B. It could have both inherited both alleles for brown eyes from both parents. All right, so it could be heterozygous or it could be homozygous. Okay, so remember, homozygous, homo means same, it means that the two alleles are identical. Hetero means different. It means that you have inherited two different alleles from mom and dad for a particular gene. One way, of course, to know if you are homozygous or heterozygous is by looking at specific characters, specific phenotypes in your parents, or say if you have been adopted, you could even know when you have children. Okay, so all right, so all these definitions are very important, okay? Genotype, phenotype, homozygous, heterozygous, gene, and allele, okay? You need to know them all for me. And remember, a phenotype is a physical manifestation of the alleles of an individual, okay? Freckles is another type of allele that shows dominant versus recessive behavior. And is a recessive allele. Okay, so phenotypes, well, we call this with this very fancy word nowadays, phenotype. At the time when Darwin was studying kind of like all these traits, was narrowing down all these observations, he called them actual with a simpler word, which is the word trait. So phenotypes are traits, okay? Or you can even call them characters. All right, so and the phenotype and the genotype can be very different. A certain type of phenotype, especially the dominant one, does not necessarily correspond to a certain genotype. For instance, let's look at some characters or that in or some genes that in human population display this kind of behavior, dominant versus recessive. For instance, hair color, dark is dominant versus blonde. Curly is dominant versus straight. And there are some other more different, more, let's say, non-obvious characters that you could test yourself. For instance, the ability to curve your tongue or not. Okay, that's dominant versus recessive. If you have hair over here on your fingers, things like that, or the widow's peak as well. Okay, it's kind of a dominant character. Eye color we talked about. So there are so many in human population. Okay, and in this diagram shows you only examples of two different alleles, two different variants, but sometimes you have more than two. Okay, more than two, and that's because there is more than one gene that determines a certain character. Okay. So, for instance, when we talk about eye color, it's a very much of an exemplification to talk about just the allele for brown eyes and the allele for blue eyes. In reality, they're still studying this. I believe they have identified between 6 and 9 different genes that all contribute altogether to the eye color. And that's why in the human population as phenotype, you don't just have blue, brown, or green eyes. You have all kinds of shades that have been recognized. Okay, and so that's also true for instance, for the alleles that determine skin color. And many times it's the combination of all these genes, or the alleles that compose these genes, that determines a specific phenotype in the organism. Okay. All right. So let's move on. Now, now that we have defined all these kind of like basic genetics terms and explain a little bit how things work, we need to understand how reproduction works. And as you may recall, we have already defined two types of reproduction in animal, not in animals, excuse me, in the living world, let's say. One is the asexual reproduction, and the second type is sexual reproduction. Remember that animals, by definition, reproduce sexually. Now, you have a couple of groups of animals that have a Plan B member, sponges, for instance. They have also the capability to reproduce asexually. But remember, as by definition, an animal reproduce sexually. At least reproduce sexually. On top of that, could reproduce also asexually. All right, so reproduction is the process by which living organisms give rise to offspring. And in asexual reproduction, for instance, we can very well here talk about protists. Remember an amoeba? Remember all those protists that we looked at a microscope this semester? I'm so glad we did that activity. Protists typically they reproduce asexually, And the type of asexual reproduction is known as mitosis. And so mitosis refers to asexual reproduction, so a type of reproduction where there is only one parent, a single parent. And as you can understand, by having just a single parent that you come from, offspring, as an offspring, you tend to be pretty much genetically identical to your parent. There is no alteration, no combination of different alleles or genes. There is only one parent involved here. So the variation in the offspring is very limited, as you can imagine. Now, one thing I want you to understand is that as an organism to produce offspring, you and all the other animals in the animal kingdom, yes, you reproduce sexually, but the cells, the majority of the cells of your body, they regenerate constantly, they regenerate organs constantly, they in fact do reproduce asexually. So you have a lot of asexual reproduction, a lot of mitosis happening in your body at any given time. It's only talking about the reproductive cells, the cells that will give rise to offspring. They undergo a different type of cell division, okay, of reproduction that is known with a different term, which we're going to introduce in a minute. Okay, so mitosis is asexual, is the type of cell division on which asexually reproducing organisms rely. As I told you, the variation in the offspring is very limited. The only chance for offspring to be a little bit different from the parent, from the daughter cells, let's say, to be different from the mother cells, is in the possibility of mutation. So mutations are random errors that may happen in the molecule of DNA when this DNA molecule replicates, as we will see. So mutations are pretty much the only source of variation for asexually reproducing organisms. All right, so let's talk about mitosis. Let's talk about the process. Here is a very schematic representation of mitosis, and pretty much that's all you have to grasp. Of course, if you have a cell that wants to replicate, and that's a certain repertoire of chromosomes, so we call it DNA, okay? Here there is not even chromosomes represented, it's just the DNA acronym. Before attempting to divide as a cell, in order to preserve the same DNA, if I split this word in two, it's not DNA anymore, it's D -N and then -A as something different. So if I want to conserve this word DNA and split the cell, this implies that I have to duplicate my DNA molecule before cell division happens. And that's exactly what happens in mitosis, where DNA is duplicated, which means all the chromosomes duplicate, to ensure that in the two daughter cells that come from the mother cell, there is the exact content and possibly identity of DNA as the mother cell. Okay? Now, imagine this DNA, as I told you, each gene is thousands of nucleotides, dozens of letters long, and organisms have thousands of genes as well. So there is a copying here, a copying of a long manuscript to be done every time that a cell wants to divide. And as you can imagine, as nature is not perfect, some errors, some even just one letter errors may happen in these very long words. And even just one letter error one letter change, which is known as mutation, may alter that the meaning of that word, and therefore the subsequent protein that is produced by referring to that long word, to that gene. And so mutations, we typically give a negative attribute to this word, right? But mutations are essential, especially for organisms that only rely on mitosis for changing over time. Okay, random mutations that may appear in a population may sometimes be beneficial for that population. All right, so again, we already talked about all this, okay? Duplication of DNA before cell division. Now, by looking at something a little bit more realistic here, okay? Instead of just writing or looking at the word the DNA that replicates, let's look at something a little bit more realistic. An organism that has only one pair of chromosomes. I think there is only one species of ant that has one pair of chromosome, and is very much of an exception. But in the animal world, you have always a few pairs, at least a few pairs of chromosomes, never such a little amount. So hypothetically, consider an organism that has only one pair. Remember, one is inherited from mom, one is inherited from dad. If we talk about animals, we talk about always diploid organisms that have two chromosomes for each representative. Okay, one is inherited from mom, one is inherited from dad. This is intrinsic to the sexual reproduction mode. So you have replication, so each chromosome duplicates, and then each of these chromosomes therefore can split after duplicate, and each representative from mom and dad will go in each daughter cell. Note that in this division, you never see two blue chromosomes going on one side and two red going on the other. The identity of that combination of chromosomes and specific combination of alleles always has to be maintained, okay? You start with a red and a blue, which represent one from mom, one from dad, and you end with the same combination in the daughter cells. You cannot put two of the dads and two of the moms. If that happened, it would be a mutation. It could potentially be deleterious. All right, so mitosis is asexual cell division and is used for asexual reproduction, but also for growth of multicellular organisms. From the time that you are conceived to the time that you're born as a baby, there's a lot of mitosis going on. We talked about this, remember, in class. But there is a lot of mitosis going on in your organism as an adult as well, from the time you are born to the time you become adult. There's a lot of mitosis going on because your body has to grow, your number of cells has to increase as well. Also, tissue repair and regeneration. If you get a scratch on your skin, how does the healing happen? The healing of your skin happens because there are stem cells that divide, and so divide by mitosis, and are going to repair that cut or that scratch that has happened in your skin. And so this is true for many types of tissues that can repair and regenerate. Some tissues don't really repair much. For instance, nerve tissue does not have capability of repair, and some nerve cells, basically some of your nerve cells that you're born with are the same nerve cells that you are going to die with. So, but many organs and tissues in your body are capable of regenerating, and they're in fact constantly regenerating. All right, so these are all the uses of mitosis. I think there is a video here on mitosis. You guys are invited to watch it. I'm not going to play it in this presentation. All right, so the second type of organismal reproduction is sexual reproduction. And sexual reproduction implies the contribution of two parents. There are only when two parents come together with production of gametes, then sexual reproduction can happen. Now, in this type of reproduction, of the offspring is genetically different from each of the parent. And there is a lot of genetic variation ensured in this type of reproduction. And that we will see what are the mechanisms to ensure this variation. That's what we have to understand. Now, so in sexual reproduction, there is a combination of genes, a combination of alleles that results from the fusion of different gametes that come from different parents. And I've already told you basically. And in sexual reproduction, the organism that reproduce sexually produces gametes through a process of cell division, which is very different from mitosis and is known as meiosis, where in meiosis you start from stem cells that are diploid, just like all the other cells of the body of the organism, and you end up with haploid cells. Remember, we talked about these terms. I hope you guys remember them. If you don't, again, diploid means having two sets, 2N is the way we indicated, two sets of chromosomes, or I say a pair of chromosomes for each set. And haploid, which is indicated as N, means having one set of chromosome of one chromosome for each set, however you want to say. So here, remember that species of different organisms or animals, but also plants, they have always typically more than two chromosomes at least, with some exceptions. This is an organism that hypothetically has three chromosomes are indicated here with different size and color. And the different size is really typical. So of the 23 pairs of chromosomes that we possess in our cells, each chromosome in the 23 of the 23 has specific size. Okay, so it is really true that they're all at different size. Again, in meiosis, you start from a diploid cell and you end up with a haploid cell. So meiosis is a reduction cell division, whereas mitosis, in mitosis, the number of chromosomes is conserved, okay, as we saw just a minute ago. All right, so, and this makes a lot of sense, right? Because in order to maintain the same exact number of chromosomes in a species, if you didn't cut in half your gametes during the formation of their formation, then, and producing haploid cells, then you wouldn't reestablish diploidy in the offspring in the next generation. You would actually double up the number of pairs of chromosome at each generation. And you know, this can never happen. Even only one extra chromosome in a pair, for instance, chromosome number 21, which is typical, the most representative of chromosome alteration in human population, determines the Down syndrome. So in the Down syndrome, the individuals have, instead of two, they have three representative of the chromosome number 21 pair. And just that little itty bit of, it's a small chromosomes, one of the smallest chromosomes in humans, determines lots of alterations. Imagine to have the entire 23 chromosomes duplicated, quadrupled. That will be impossible. That will be not permissive for life. So the number of chromosomes must be maintained finely regulated and constant in offsprings at every generation. All right, remember meiosis happens in the reproductive organs, of course, in testes in males, if we talk about humans, and in ovaries, if we talk again about, this is humans represented here. But basically, these terms are used pretty much also in plants. Also in plants, you talk about ovaries, oviducts. All these terms are pretty much very, very universal. All right, so how do you get from a diploid cell to haploid cells during the meiosis division? The trick here, first of all, remember that in each cell division, you cannot obtain more than 2 cells. In other words, a cell can only split in 2. It cannot split in 4 or in 6 or in 8, okay? So the trick, therefore, here, in order for meiosis to reduce, to cut in half the number of chromosomes, is that you can have 2 consecutive cell divisions. And in these 2 consecutive cell divisions, you start from 1 cell, you divide it in 2, and then each of these cells divides in 2 again, and you end up with 4 cells. Okay, so let's start looking at the big picture here, where you go from 1 cell, which is diploid, to 4 cells, which are haploid. And by doing so, you're able to cut down your number of chromosomes in half. Now, this cut of number of chromosomes in half is not random. It is a very precise process in which, let's see what's happening here. I like this diagram because it's simple enough for you to understand what's going on. First of all, mom and dad chromosome are always represented with 2 different colors. Remember, in each of your stem cell that gives rise to, let's talk about sperm, because for eggs is a little bit different. I like to talk about sperm formation because it's the typical example where, for instance, in a male, in the testis, you have a repertoire of stem cells that constantly divide, that are diploid. Of course, you start from a diploid cell. They're just like any other cell of the body of the male. But because these are cells that will give rise to gametes, they undergo this process of meiosis to cut in half the number of chromosomes to give haploid gametes. And so this sperm cell has 2 representatives here, one from mom, one from dad. Before attempting the first meiotic division, these 2 chromosomes duplicate, just like it would happen in mitosis, right? So these are duplicated chromosomes. And after that has happened, this cell divides and splits the number of chromosomes in half. Now, I invite you to look at what's happening here. In this division, this is very different from what we have seen in mitosis. Here, you have that one cell receives the chromosome that male had received from mom, is duplicated, and one cell receives that chromosome they had received from dad. And these pairs of chromosomes that you receive from mom and dad are called homologous. Homologous pair because homo means the same. So they are chromosomes that bring the same sets of genes, although they could be different gene variants, different alleles, but the sequence of genes that you find on this chromosome is the same exact sequence that you find on this one. For instance, if over here there was a gene that determines the eye color, that would be true also for the homologous. Now, it could be that this one is the allele for brown and this one is the allele for blue. Yes, but it's the gene that determines the eye color. And so, during the first meiotic division, you first duplicate your parental chromosomes, and then you split them. You sort them out. Okay, let's say as duplicated. In the next cell division, now, which resembles very much a meiosis process, you're going to split the duplicated chromosome. What are you ending up with? With 4 haploid cells now. Each haploid cell only receives one representative of the pair of chromosomes that you have inherited from mom and dad. In other words, think about yourself. Okay, if you have already reproduced, if you already have children, what this all means is that your children, look at your children if you have children, or think about having children if you haven't yet. This means that you, in yourself, okay, you have the chromosomes that you have inherited from mom and dad. Now, what you will pass on your offspring is one of the set of allele variants that you have, of gene variants or alleles that you have received from mom and dad, respectively. So, if this were your gametes forming right now, at this very moment, if this is the sperm cell that will fertilize the egg of a partner, of a mate, then this means that that offspring, that person, could be a boy, could be a girl, will receive the gene variant or allele that you had inherited from your mom, okay? Whereas if this was the sperm cell that had fertilized the egg of a mate, now in the offspring, you have that child or that organism, whichever species you want to consider. It's easy to think about yourself, okay? The way you work, your body works, I think, in this regard. It's you are able to understand a little bit better if you relate it to yourself. So this means that that particular offspring, if this is a child that is born, then that child will receive the variants of the genes or all the alleles that you had inherited from your dad, okay? So in other words, in your offspring, you either, for each chromosome, you either give the one that you have inherited from mom or the one that you have inherited from dad, okay? Now, this is a very much of a simplification, because in this diagram, it shows you only one pair of chromosome, but you know, you have 23. And of course, I'm not going to present you the scenario of 23 pairs of chromosomes, because otherwise next year, this time, we're still talking about it. But what I do want to show you is a little bit more realistic situation where you actually start maybe with 2 pairs of chromosomes. And here the 2 pairs of chromosomes are represented with 2 different sizes. Okay? So, okay, let's analyze what's happening here. Now, don't get confused, don't get frustrated. I see my students, when I start talking about these things, they get a little lost. I always tell you guys, and now you are at home and you are able to do that. If you happen to have conserved the pieces of Legos from when you were younger, do this for me. Just take 2 pieces of Legos that are 2 different colors and 2 different sizes as well. okay? But each of the different color has to have the same size, okay? Just like represented here. And you know, with pieces of Lego is easy because you can take two blue pieces that are the same size, and fuse them together. You know, attach them together, so you can, and then you can also separate them very easily. That's why playing with Legos is really good example. So imagine I asked you um, to separate each of the these pairs of chromosomes, to where at the end of this separation, okay, you must have only one of the fused part, okay, let's say of the attached part, but you must have each representative for each size, okay, which is what you're seeing here, okay. For instance, in this diagram, it shows you that you have done this separation where you have separated the attached pieces into single pieces, and you have respected the rule that I told you, because you have given me each, you have separated them by size. So each of this combination has a long and a short size, okay? But here is the dilemma. If I tell you to do that, maybe I give a group activity. I'll guarantee you that some students will have this combination and some students will have this combination, because once you have separated these two big ones, now the dilemma is, do I put the small blue with the big blue, or do I put the small red with the big blue, as shown here? So it's a dilemma, and the possibilities are 50-50, in other words. And so as the possibilities, these combinations are up to you, and if I gave you an assignment in class, probably what the result will be 50-50, just by random chance, I would imagine. So it happens in nature. In other words, you have different possibilities, different combinations that can happen of assortments of the chromosomes that you have inherited from mom and dad during the formation of your gametes. And starting with two chromosomes, two pairs of chromosomes, you end up with 4 different combinations, as shown here. And each combination can happen independently from another, okay? It's just a matter of chances, 50-50. So chromosomes can independently assort. And this phenomenon that happens was initially understood indirectly by a genius of the father of genetics, Mendel, Gregory Mendel, and he stated, I was able to understand how things work during formation of gametes, and he called these different combinatorial factor as law of independent assortment, which states that, again, in the formation of gametes, the maternal and paternal chromosomes that an individual has inherited will independently assort, okay? If this big one and the small one were inherited from dad, they have no link with one another. There is no reason why all the chromosomes that you have inherited from your dad should go all together with one another, and all the chromosomes that you have inherited from mom should go together with one another. There is no reason, there is no link between these two different chromosomes. So the assortment can be equally possible, okay? And so in other words, there is and consider another thing. The higher the number of chromosomes in a species, the higher the combinations that can happen. And I'll tell you in a minute how to come up with the number here. What is that mathematical operation that gives you the number of combinations? Okay, they starting from a specific number of pairs of chromosomes. And so, with that said, that operation that you have to conduct is 2 exponential 23. So remember, we always talk about diploid organisms here, animals. I'm sorry, 2 exponential x, I should say, because here I'm talking about our species, our human species, but if I am a mouse, this will become 2 exponential 20. So 2 exponential, the number of pairs of chromosomes, it's the number of combinations of gametes that an individual can produce during the formation of gametes. So in humans, because we have 23 pairs of chromosomes, the number of combinations in each of us, the number of combinations of chromosomes that can happen in our gametes of the chromosomes that we have inherited from our mom and dad is given by this operation, and this is more than 8 millions, 8,300,000 something. You can do it with a calculator. So this means that there are more than 8 million different types of gametes that you and I can produce given the chromosome that we have received from our parents. Okay? And of course, because we reproduce sexually, every time you reproduce means that your gametes will fuse with the gametes of a mate. You have to multiply these 8 million or 8,300,000 something by the 8,300,000 something of the mate. And this will give you more than 64 trillion combinations. In other words, if the same individuals reproduce over and over, a man and a woman, they marry each other and have hundreds of kids, well, that's impossible because a woman can only, produce so many kids during lifespan. But theoretically, imagine if a couple would have hundreds of kids, the chances that two of their children having the same combination of chromosomes is very, very, very low. It's less than 1 in 64 trillions. So this is a genius thing, right? That nature has created to ensure that all individuals are different from one another, to ensure difference and variability in a population, even a population of offspring that comes from the same 2 parents. Imagine insects that have a very high reproductive capability, as we said, this is one of their strong adaptation for success. Imagine that a female of an insect can deposit hundreds of eggs at any given time. And so imagine that there is a male mosquito, the same male mosquito that somehow fertilizes those eggs, somehow mates with that fly or mosquito, whatever it is. And imagine that in that regard, in that case, the numbers are much higher. Okay, the number of offspring is much higher. So again, to ensure the variability in those populations, the number of chromosomes, that's what determines the higher number of chromosomes, the higher the number of combinations and the variability in the offspring. All right, I know, guys, this is a little bit tricky probably to understand, but really, if you clear your mind, it's not that difficult. All right, so now The one thing that I didn't tell you, I presented the process of meiosis. Remember, there are two consecutive cell divisions where the homologous chromosomes first divide and then they split. Here, the homologous, again, there is a cell in which, in a hypothetical cell of an organism in which there are only two pairs of chromosomes. The moms are represented in red and the dads in blue. And there are two different sizes, two different chromosomes. Typically, chromosomes do present different sizes. They duplicate, of course, and then they split. What you may notice here is that the duplicated chromosomes here, something has happened to them. There has been a swap of pieces of chromosomes from the chromosomes that this individual had inherited from mom and dad, respectively, right? So this chromosome is not completely blue anymore, and this is not completely red anymore. It seems like the red piece now is over here, and the blue piece that was over here now is on the red. So there is a swap of pieces of chromosomes, and this swap is known as crossing over or recombination. And is another mechanism that again ensures variability, because now this crossing over is very random. It never happens the same in any of the cell divisions that happen during the formation of gametes of an individual. And so again, it represents an additional way to create different combinations of alleles. Okay. So crossing over is another mechanism to generate variation in offspring. Here is another video. This is a video about meiosis. Again, I invite you to watch it if you have time. It gets a little bit technical. Don't worry about all these, defines you all the phases of meiosis. Of course, here we're just summarizing the process because we want to grasp mostly the meaning and the results and the significance of this process to look at it in a bigger picture. Okay, so we're not going to go into the details of these processes, neither mitosis or meiosis, but the video does give you these more technical definitions. Don't worry about those. But I do invite you to watch it because it really shows you how the division of the chromosomes, the cell division and the split of the chromosomes happen. All right, so independent assortment gave us a lot of variability. Crossing over does give us even more variability. Okay, this is independent assortment, and this was the crossing over. But we know that there is another element here that can always happen, another element of change, because remember, no matter if you're doing mitosis or meiosis, before a cell division, the DNA must be duplicated. And that event itself is an event that has an intrinsic error, possibility of error in it. In fact, I guarantee you that at every cell division in your body, there is always a little bit of a mutation, at least one mutation that can slip in. Unfortunately, sometimes these mutations happen in important genes, and that's what sometimes causes cancer in people. So to summarize, in sexual reproduction, genetic variation in sexually reproducing organisms, genetic variation is ensured by three different mechanisms. The first and foremost that produces has more weight, let's say, on the variability of the offspring is the independent assortment of the homologous chromosomes, which are the chromosomes that are inherited from mom and dad, from the two parents. The second level of variability is ensured by crossing over or recombination. Remember this swap of pieces of chromosomes between the homologous chromosomes during the formation of gametes. And the third line is mutations. Remember, mutations are rare in living organisms that are much more frequent in things like viruses. Viruses have the luxury to mutate very, very quickly and a lot because they have a different strategy of survival. And I'm quoting the word survival because viruses, in fact, are not even considered living, but their strategy is to mutate so quickly because they can produce millions of copies of themselves in every cycle of in every reproductive cycle. Therefore, even if there are many deleterious mutations, that doesn't disrupt them. But amongst all these mutations, there will be some that make them stronger. And so they will perpetuate stronger and stronger over time. All right, so this slide is very important. You must be able to rank all these phenomena that are responsible for genetic variation in sexually reproducing organisms for me. Okay. All right, so guys, I think again here there are summary slides that reinforce what I told you. Humans have 2 sets of 23 chromosomes, so more than 64 trillion possible offspring of 2 parents that mate constantly over and over. And of course, this ensures again that no 2 siblings are ever going to be identical, at the exception of identical twins, but identical twins are represented from a split that happens in the embryo after the first mitosis, to where then each cell becomes an embryo. Remember, we talked about all the stages of the early stages of embryonic development. There is a first cell mitosis that happens in the embryo, but that's a different story. All the combination of chromosomes has already happened in an individual. So the identical twins have the same exact set of chromosomes, and they have also undergone the same exact event of crossing over. All right, so recombination. Remember, so first independent assortment, secondly recombination, and thirdly mutation. Now, talking about mutation, just in a little bit more detail. Mutations, remember, are random and accidental. They're just totally random. Remember, if I am copying an essay that is really long, I may get tired and make some typos. These typos represent my mutations, okay? They're very random. There is no reason why it should happen in one place more than the other. And some are really small. Some mutations can be larger. For instance, sometimes a portion of a word may be completely missed, or just a letter could be missed, or a letter could be introduced, or sometimes an entire sentence maybe could be deleted. So it depends. Sometimes mutations can be large. And we talked about this semester about one particular type of anemia, if you remember, sickle cell anemia, that changes the shape of a red blood cell from round, kind of like a disc in reality, to a half moon. And this sickle cell anemia is determined by a very small mutation. There is only one change in one letter of the gene that codifies for hemoglobin. Okay, only one letter, okay, in that long word, and you end up with dramatic changes. If you happen to inherit this mutation from both mom and dad, so you are homozygous, you cannot survive further than a couple of years of age. so far. And I think they are making a lot of progress in gene editing for people that carry this trait, this mutation for sickle cell anemia. All right, so mutations can be also really big. For instance, some children may be born lacking an entire piece of a chromosome. Again, there has to be a balance here. If it's too much, maybe that is not permissive for life. But for example, this is a mutation that deletes almost an entire arm of a chromosome number 5. It cuts it in half. This mutation is known as Cri-du-Chat. It gives lots of problems and a specific, a very typical facial characteristics, mental retardation, and it's called Cri-du-Chat, which means cry like a cat. It's from French. And the typical thing that these children present is that at birth, instead of crying like a human being, they sound like a cat, totally like a cat. That's why this is called the Cri-du-Chat syndrome, the syndrome to cry like a cat. And it's just one of the characteristics, but not the deleterious or the other more deleterious characteristics are skeletal imperfections or deformations and also mental retardation. Okay, and it's an entire piece of the chromosome number 5 that one of the chromosomes is absent, as somehow been lost during the formation of the gametes that then gave rise to these individuals. Okay, that can happen. It's kind of a mutation. All right, so and remember that as much as you cannot avoid errors in the process of copying of DNA during the reproduction, either mitosis or meiosis, so either asexual or sexual reproductions, because remember, both processes imply DNA doubling, DNA copying. There are intrinsic natural errors that can happen during the copying process that you cannot do anything about. But there are many agents, physical or chemical, in nature that can increase those rates of mutations, can directly damage DNA. And when the DNA is damaged, basically it's like, imagine you are copying an essay and you spill coffee on the paper that you're trying to copy. And that coffee masks a word or partial word. Now you're trying to guess what was written there. You have damaged your template. If you damage your template, now maybe you're going to make some errors because your template is compromised. That's the same concept. If you damage DNA of a cell during the copying process, because there is some damage, the system tries to guess and maybe it doesn't copy what exactly was the template, the meaning of the template. It copies something a little different. That is a mutation. So there are agents in nature that increase damage in your DNA. These are, for instance, UV light or many chemicals that come from cigarette smoking, okay, or radiation. Radiation is a big one. And we have learned this very harsh way when there was. I'm just going to show you this one first, and then I'll go to the previous one. I don't know how many of you know about Chernobyl, where there was a nuclear implant that exploded. I was a preteen at this time. Now you know how old I am. And basically, actually, I was a teenager. No, kind of almost. So, anyways, I remember I was living in Italy, and this wasn't very far from where I live. This was in Russia. And many children were born with malformations during that time because a lot of radiation was released in the environment. This radiation affected lots of people, and when you affect especially a developing embryo with mutagenic agents such as radiation or chemicals, this developing embryo is much more susceptible to malformations than an adult, of course. And all these damage that you are putting on DNA are going to be they are going to result in a lot of damage to the organism. Okay, now again, there are some chemicals that you have to try to stay away from. One of them that many people give to themselves by choice is there are all those chemicals that are present in cigarette smoke that over time they damage your special lung DNA, bringing eventually to the possibility of developing lung cancer. Because if that, again, there are some genes in our body that are very important to defend ourselves against cancer. And if you damage those very genes, then you're out of luck. Cancer is going to arise. But as much as that is true, maybe a smoker has more chances to develop cancer because of those chemicals being mutagenic. In reality, the majority, close to 70% of cases of cancer cases that arise in human population every year are in reality the result of the intrinsic error that happens during DNA replication. In other words, even not exposing ourselves to mutagenic agents necessarily, we still have a good chance of developing cancer during lifespan because most of cancers are the result of that intrinsic mutation rate that is typical and happens anyways during the copying process of DNA. All right, so the only way we have in our lifespan to try to prevent or try to intervene when cancer arises is to either do cancer prevention or so try to stay healthy. Of course, you don't want to increase your possibilities of developing cancer, but also doing our screenings, our annual screenings after we turn 40. All right. So it's interesting to talk about mutations, and mutations happen in all organisms, not just in the human species. All the kind of mutations that happen in our cells in our body, in gametes, regarding gametes or regarding your other cells of the body, which are called somatic cells. They happen in all other organisms, plants, animals, without any distinction. All right, so I think I'm going to stop here for this first part of the lecture. I'm going to take a little break, and then I'm going to generate a part 2 of this lecture where we're going to talk about allele frequencies in populations. And if you have the chance to study well this part and then move on to the second part, that will be the ideal thing to do. Okay.